So the process by which DNA is converted into RNA is called transcription. And the overall process of transcription is mediated by three different steps. The first one is initiation. The second is elongation. And finally, termination. In this video, we are going to specifically talk about the initiation process in bacteria or prokaryotes. Before we move into the initiation process, first we need to know that the whole process of transcription is mediated by an enzyme called the Rna Polymerase. Now, this RNA polymerase, there are different kinds of RNA polymerase, but in bacteria, there is only one type in bacteria or in prokaryotes. Whereas in eukaryotes, there are different kinds of RNA polymerases, like RNA polymerase 1, 2, 3, 4, 5, and so on. But in bacteria, there is only one kind of RNA polymerase. Now, this RNA polymerase has got 4 different subunits that make up the core enzyme of the RNA polymerase. So, the core enzyme is made up of 2 alpha subunit, 1 beta subunit, 1 beta prime subunit, and 1 omega subunit. These comprises of the core enzyme, and if the core enzyme is added on with a sigma subunit, then the core enzyme along with the sigma subunit would be called as the holoenzyme RNA polymerase. Now, this sigma subunit has got a specific function. That is why it is required. This sigma subunit is actually required to recognize a certain position in the DNA for the RNA polymerase to bind, and this region is called the promoter sequence. Now, the promoter sequences are present in every gene. Every gene has got a promoter sequence so that the RNA polymerase can bind to that sequence and start the transcription. Now, if we take a look at the DNA, and let's say this is the gene, then the first nucleotide from where the functional RNA needs to be transcribed, let's say over here, the first nucleotide from where the functional RNA of the gene needs to be transcribed, that is referred to as +1. And when we go towards the 5' end from this +1, that is towards this side on the left, the number becomes negative. That is -1, -2, -3, and so on. Now, the promoter sequence is somewhere near the -10 and somewhere near the -35. These two sequences are recognized by the sigma subunit, that is the holoenzyme of the RNA polymerase. Now, around the -10, the sequence is something like TA-TA-AT. And around -35, the sequence is like TT-GA-CA. And these two sequences are called consensus sequences. These consensus sequences, these are generally conserved in different species of bacteria. So these two regions, -10 and -35, these are recognized by the RNA polymerase. And there is sometimes another sequence which is called the up element, which is upstream of -35, and this up element is generally present in the genes that are encoding the ribosomal subunit. So the RNA polymerase scans the DNA, and the sigma subunit recognizes and binds to this region pertaining the -35 and -10 sequences. Now, when the RNA polymerase binds along with the sigma subunit, this whole complex is called closed complex. Now, when the RNA polymerase would start to synthesize RNA, then what happens is that the DNA would sort of denature a little bit. The DNA would denature a little bit, and the RNA polymerase, along with the sigma subunit, it would synthesize a little bit of the RNA. Now, the RNA polymerase can only synthesize RNA in 5' to 3' direction. It cannot do in the reverse manner. So if this is 5', it's 3', 5', and 3'. So the RNA polymerase would start synthesizing according to the template strand in the 5' to 3' direction. Now, when this happens, when the DNA unwinds a little bit and a little stretch of the RNA is synthesized, this is called the open complex. So closed complex is when the DNA is not unwounded, and the open complex is when the DNA starts to unwind and a little stretch of the RNA is synthesized. And as the RNA polymerase moves along the DNA and has synthesized at about 10 nucleotides of RNA, what happens is this sigma subunit falls off. So this sigma subunit falls off, and the RNA polymerase can move on to synthesize the RNA of the complete gene. So this sigma subunit is only required to recognize the -35 and the -10 region, or specifically the promoter sequence. One more thing is that the RNA polymerase does not require a primer for starting the synthesis of RNA. So it does not require primer, or it is primer independent process. And the RNA polymerase can synthesize the RNA from scratch, that is de novo synthesis of RNA by the RNA polymerase. So it does not require primer. This is a very important point. And that is all about the prokaryotic transcription initiation.